Okay, so today I'm going to give you an overview of the research that is done in my lab. I'm Dr. Klauda. Come from the Chemical and Biomolecular Engineering Department, but I'm part of the Biophysics program. And I have 5 students, graduate students, that are in my lab. One of these is from the Biophysics program, Yalun Yu, and I have Madi and other students that are from chemical engineering and one from chemistry, and a bunch of undergraduates. And so they all look pretty happy. I think they're happy to be in the lab and to do the research that we are doing. So what do we do? So what we do is to take and look at research at a multi-scale approach. So if we're looking at these proteins here that are involved in drug transport, specifically maybe efflux proteins that are involved in antibiotic expelling of antibiotics from bacteria, that if we want to understand how the drug is expelled from bacteria to better design drugs to actually kill bacteria, one option, we really need to understand the mechanism of binding as well as export. And so that we could potentially design drugs that do not bind to these proteins, or also might be a co-drug that binds to these proteins and deactivates them so that the actual drug kills the bacteria. And so the research we do is computational. And so we don't have a wet lab. We use computers, high-performance computers, to do our research. And we have resources on campus, DeepThought 2, which will be upgraded to DeepThought 3 hopefully this summer. We have access to a joint resource at Johns Hopkins that has reasonably upgraded GPUs, and then also resources at National Supercomputers and a resource, Anton 2, that just does MD simulations. And so students in my lab get access to these machines, get experience in actually doing computation with running simulations on multiple nodes. And so we can run simulations on hundreds of cores, thousands of cores if needed, or multiple GPUs to get the desired output that we need. And so a good majority of my lab focuses on cell membrane research. So we look at various aspects of the cell membrane, whether it be a protein that resides in the membrane that transports water, like channel proteins or ions, membrane transport proteins that I mentioned before that transport drug molecules like antibiotics, to ones that have large extracellular domains that act as signaling proteins that might send something outside the cell and send a signal into the cell. But we also look at proteins that just bind on the surface of the membrane and are involved in various processes such as lipid exchange. And so the bilayer is fairly complex. We have proteins, we have lipids, and the picture here shows kind of a unified lipid structure of a bilayer. But in fact, the complexity of this bilayer in terms of lipid diversity is quite strong. The lipids in one part of the cell are much different composition-wise compared to, let's say, the plasma membrane compared to the endoplasmic reticulum. And so part of the lab is to understand why that's the case. And so one aspect in the lab is to focus primarily on the lipids. And so Yalun, his work is working on developing and improving upon our force field that describes the interactions between between lipids in our simulations. And so he's developing a new algorithm, a computational algorithm, to predict how a change in parameter will ultimately affect many different properties. We're using this to then eventually test different force fields and applications to model organism membranes like yeast, bacteria, as well as mammalian membranes like those in the ocular lens, neurons, and lung, as well as membranes that are associated with the encapsulation of the virus, RNA. And in addition to that, we do some research on specific projects that are of interest to students. And this example here is we're looking at a lipid that forms pores that could potentially be used to deliver drugs. And so one potential project that currently is available is one in which we're looking at metal-ion binding. And so this is a joint project with Sergei Sukarev's lab in biology and also part of Biophysics, the leader of Biophysics, director. This is a project started originally by Allison, who was a biophysics student, who is now a postdoc at the University of Delaware, and Yuxing, who is continuing some of this work. And so what we want to understand is how beryllium, its ion form, is toxic to cells. This causes a lung berylliosis. And what we believe the reason for that is that there's an inflammation due to the fact that the ions are affecting the pathway of getting rid of dead cells. So when cells are dead, the negatively charged lipids expose to the outer surface of the membrane, and then there's these proteins that recognize this and develop a cascade to get rid of dead cells. What the beryllium does is affect the natural calcium intermediate between the protein and the membrane, and then affects the pathway, and that's what we're trying to understand. And we need to develop a better force field for this, likely with including polarizable effects, and then do experiments in Sergey's lab. We've also recently been interested in looking at the stratum corneum, specifically the lipid structure of the stratum corneum. And so this is the outer layer of skin. This is the layer that you would touch with the environment. It consists of dead cells, as well as stuff between these cells that look much like a mortar. So the dead cell is a brick, the lipid phase is the mortar phase. This is not a typical bilayer structure. It contains fairly complex lipids that self-assemble to something that's different from a bilayer. And so we've been trying to model this and compare with experimental data on these lipid mixtures to eventually look at understanding the lipid structure in the stratum corneum, which is the main resistance to permeation of drugs across the cell, across the stratum corneum. In addition to membranes, we do work with membrane-associated proteins, and so we've been doing work with channel proteins like the aquaporin 0 protein, which is involved in early onset cataracts, drug transporters I showed before with antibiotics, and then these peripheral membrane proteins that bind on the surface. Okay, so focus your attention on the bottom half of this slide, and we're going to be looking here at, switch this to standard. And so we're going to have two movies here of this peptide that's interacting with the membrane. This is part of a larger protein that's involved in lipid movement. This peptide has a two-phase character, a Janus character, one in which one side likes water, one side does not. And so we did in our simulations is just place the peptide in the water phase. The waters here are just not showing it. And then just let the natural forces drive it towards the membrane due to attraction and see the mechanism of binding. Okay, so this is sideway view of the membrane. This is looking down on the surface. The white color is carbon, doesn't like water, red and blue like water, oxygen and nitrogen. Okay, so these movies are the same, synchronous movie. So initially the termini gets in contact with the membrane and then slides itself down into the hydrophobic pore. If you look from the top, those blue and green residues are facing you, and then the white residues are into this green, forming this membrane pocket. And so we're able to use this technique to look at, how does this peptide bind, eventually look at binding of the full protein to look at lipid exchange. So ultimately, we're not forcing this in these simulations, but ultimately we're looking at understanding how and the mechanism for binding of this peptide. And we're also using these techniques to look at other ways of binding for other peptides like antimicrobial peptides. Another aspect in the lab has been to work and collaborate with NIST, and so we've been doing work with NIST and looking at developing measurements for biomolecules, and so specifically looking at developing a field effect transistor and having a joint project by doing experiments in NIST as well as doing simulations on proteins in which we will be doing measurements of. And so let me go back and switch to laser pointer. And so what we're doing here is to look at this serotonin receptor, measure serotonin binding through simulations, and then also experimentally measure this through measurements with lipid-bound protein as well as proteins that reside in the neuron itself. And we've been kind of progressing more because of computation to larger systems, and so we've also been interested in this plexin A protein, which is involved in signal transduction. A large portion of this protein is outside the cell, and it complexes with other proteins, neutrophilin and sema, to promote and indicate cell growth. And so understanding cell growth has got a wide level of implications. Understanding growth on the neuro side, or bone growth, or cancer, and we're looking at specifically plexin A1 here in the future to look at how pancreatic cancer can progress with this protein. So we're looking at the association here as an important tool. Once we know the association, we can then potentially develop inhibitory peptides with my collaborations at the University of Virginia through Brian Berger to understand how to inhibit dimerization and then inhibit growth and potentially inhibit cancer. The current project that we're doing is looking at lipid cholesterol exchange, and so typically it was believed that lipids were moved from inside the cell to outside to the outer portions of the cell through a vesicular pathway through cytoskeleton tracts. However, if you remove that pathway, there's still 80 to 90% lipid movement. More recently, over the last 8 years or so, it's been shown that membrane contact sites are driving the motions of lipids. And so that's what we're looking at here, trying to understand how the protein actually facilitates contact sites as well as lipid exchange. And then finally, one area of research that doesn't involve membranes is one where we're looking at a protein called a polysaccharide lyase. These are enzymes by bacteria to reduce down polymers. It's important for the stability of bacterial biofilms, and so potentially utilizing this to eventually develop a better antibiotic. We're also, because I'm an engineer, we're also thinking about ways to use this as a way to develop a better enzyme to convert starch from feedstock like corn to developing a biofuel or biochemical. And so we're trying to understand the mechanism of this protein that is very pH specific to sugar cleavage. Certain sugars cleave at different pHs, and so we're trying to understand that, again, through collaborations through Brian Berger's lab. So I try and give you a very quick overview of the research we do. Where have my students gone? My students have typically go to postdoc positions afterwards, and so my first student did a postdoc at San Francisco, University of Pittsburgh, and then currently is now at a company, Pfizer, in Singapore. Other ones are currently in postdocs or in industry. A more recent graduate directly went to industry, AstraZeneca. And Viviana has a faculty offer and will be accepting this at the University of Buffalo. So if you're interested in industry, if you're interested in academia, my lab has many examples of going to those tracks. And so ultimately, we like to publish. These are some examples of cover articles of our publications over the many years. Feel free to contact me with my email shown here, or go to my website where there's more information on the research we do. I look forward to hopefully seeing you, accepting our offer to University of Maryland, and seeing you come in the fall. Thank you.